Good morning. Today is Wednesday, September 29th. We are on to our third section of 430, and that is going to be the skeletal system. Pretty straightforward schedule, although things are going to be a little bit different. If you noticed up to this point, because of the histology, because of the other things, we've been kind of combining the lecture and the lab together. So now we'll really get the opportunity to split the material up, to really have a lecture component and a lab component for that kind of thing. And to make it a more interactive process, as we've talked about, I'm going to have you guys do the bones and bone features. We are going to go through them together to make sure that we at least are exposed to them. And I get to stand here where the bone and go, this is the greater trochanter, and this is the lesser trochanter, and this is the, condyle and all of that. And it would be incredibly boring. So it's a little bit more interactive if you guys are the ones who are doing that. So that is what we're going to be doing today. Today, we are going to start our introduction to the skeletal system by focusing mostly on the histology and the base components of bone connective tissue. And then we'll be breaking up into groups where you will get assigned a bone or bones that you'll be responsible for presenting the bone features on. And we'll talk about that as well. We'll do that twice for the axial skeleton and also for the appendicular skeleton. And while it is a graded assignment, you are not being graded on your presentation skills. So if you want to, stare at a piece of paper or have your notes written on your phone and read it off your phone while you're doing that the whole time, I don't care. It's about just making this a more interactive process. And like I said, we'll talk more about that after we get through the lecture today. There are 4 assignments in this section. On Wednesday, the 6th, your first unit review is due. On Monday, the 11th, your second unit review is due. And on Wednesday, the 13th, your third unit review is due. So unit reviews 7, 8, and 9 are due. And you've done those before, you know what to expect from that. However, also on the 13th, and notice it's the last possible day that it could be due, is a 30-point skeletal review. Now, again, this is going to be something that is graded for correctness. And when people see something worth 30 points and that when they get something wrong, they lose points for it, they want to start working on it right away. But hopefully, as you've seen by looking through the modules, it's not available yet. And that is on purpose, because as I mentioned, what we're going to be doing for the next couple weeks is staring intimately at each little individual bone, learning all of its nooks and crannies. But it is important to remember that these bones come together to form your skeleton. So we need to understand how bones relate to each other. We need to understand how the trochlear of the humerus fits into the trochlear notch of the ulna. And the problem is if you dive into that right away, it's not going to make any sense. Because if you don't know what the trochlea of the humerus is, if you don't know what the trochlear notch of the ulnar is, then it's not going to make any sense trying to put those pieces together. So I purposely have not allowed it to be available for you yet because I don't want you thinking about it. I don't want you working on it because it would be too challenging at this point. As you start to learn the bones and bone features, that's when it'll be easier to understand how the pieces fit together. And that's what that review is all about. So you'll have some time to work on that. But first, right now, I want you to focus on learning the bits and pieces of the bone, and then you can put them together to make a skeleton. All leading up to the 18th, which we will then have our lab and lecture exam. So that is the game plan. Any questions on that?
I do have a question.
Oh, yeah.
Are we going to still go over the cancer?
I'm sorry?
Are we still going to go over the skin cancers?
No, we've moved beyond cancer. We've moved beyond the skin, so you will not be responsible for that information. Obviously not on the exam because the exam's done, but you will not be responsible for it on the final. So again, that's material. We ran out of time. We didn't get a chance to cover. Your book, if it's something that is of personal interest to you, your book is a good place to start. It does do a nice job about talking about them, but it's not material you'll be responsible for on the exam, on the final. And obviously it wasn't on the last exam. All right. Any other questions? Yeah, like I said, it would be nice to go over those things. We ran out of time. And that happens sometimes. All righty. On to the skeletal system. Skeletal system comes from the Greek word skeletos, which means dried up, because normally when we think of bones, we think of something like that, right? When we see them out around Halloween, they're these dead, dried up pieces of tissue. However, when they're actually inside your body doing their job, they're actually incredibly dynamic tissue. If you remember, we were talking about this before when we were talking about how well vascularized they are, which allows you to break your femur and have it heal in 7 weeks, as opposed to tearing that cartilage, which may never heal properly again. So as we'll see, bone is an incredibly dynamic tissue with a whole lot going on with it. So it's going to be an important and fun thing to talk about. It is, of course, a skeletal system, an organ system, which means it needs to be made of organs. What are the organs? Are there really organs in the skeletal system? What do you think? Yeah. What are the organs?
Is it the bones themselves?
Yeah, the bones themselves, absolutely. Bones are indeed an organ. If you think back to our definition of what an organ is comprised of 2 or more tissues working together to serve some function. Yes, bones are primarily made up of bone connective tissue, but there's also like the hyaline cartilage that we know that's on the edge of the joints. There's nerves and blood vessels passing through them. They absolutely are organs. And how many of them do we have?
Like 206 or something.
So it turns out we have, every single person in this class has far more than 206 bones in their body. However, that number 206 is something that comes up all the time because we have 206 named bones. Right? These are bones that are actually identified by a name. We also have a bunch of additional bones that are small and irregular and differ from person to person. And so you're not able to give them names. So absolutely, there are 206 named bones in the body. And guess how many of those you're going to have to learn?
All of them.
All of them, absolutely. But the good news is many of them are paired bones. What does that mean? True, they do work together, but when we use the term paired in this case, that's not really what we're referring to.
There's 2 of the same ones because like you have like a right hand and a left hand usually.
Yeah, exactly. By paired in this case, what we mean is that you have a right version and a left version. So you have a right humerus and a left humerus, right? So the good news is once you learn all the bits and pieces about one humerus, it's the same bits and pieces on the other humerus. However, with our paired bones, you will need to distinguish left and right. So when I show you a picture of a bone that is a paired bone, you are going to be responsible for being able to tell whether that bone is right or left. But again, as we're working together in the groups, that's what you're going to help us to do. Excellent. And then, of course, the study of bones is the field of osteology. And of course, the study of the movement of those bones is the field of kinesiology. All right. We have talked a little bit about the skeletal system already. And when we talked about it before, we identified functions of the skeletal system. And what were the big 3 functions that we talked about? What's a function of the skeletal system?
Support.
Movement, excellent. Support and protection, absolutely. Those are the big 3, right? Again, they form the levers that allow our muscles to pull on them to be able to move our body through space. They give us our vertical and our horizontal axis of our body, allowing us to stand upright, and many of our organs are protected by bones. So absolutely, those are the big 3 that we talked about before. However, there are other important functions of the skeletal system as well. One of them is hematopoiesis. Any idea what that is? Hema. What does hema remind you of?
Hemoglobin.
right? And where is that found?
In our
Blood. Absolutely. Our skeletal system is responsible for making all of the formed elements of our blood. What were the 3 formed elements again of our blood?
Is that the leukocytes, thrombocytes, and erythrocytes?
Yeah, exactly. Absolutely. Or what we commonly call the red blood cells, the white blood cells, and the platelets. Absolutely. Right. So all of those cells are made where in our bones?
Bone marrow.
Although, can we be a little more specific?
central canal.
Okay, well, remember, the central canal is where blood vessels and the nerves are located, but there we go. That was what I was looking for. Red bone marrow. All right. Of course, that should be a big hint right there. If I say red bone marrow, the implication there is that there's another type of bone marrow. Excellent. And so perfect. So we have that ability to make all of our formed elements. Anytime we need more red blood cells, white blood cells, or platelets, we can do that in the red bone marrow. Our bones also play a very important role in maintaining homeostasis of many of the minerals in our body. This includes primarily calcium. Something like 99% of the calcium that is found in your body is located in the bones. And as we've already talked about, calcium makes cells do wonky things. So calcium is very, very important to maintain in our body. But there's also phosphorus and manganese and all sorts of other minerals that are located within our bones as well. Now, where in our bones are these things located? Nope, not the white, not the red bone marrow. It is in the white part, but the white part isn't bone marrow. What do we call the white part of the bone?
Compact?
True. The white part is compact bone, and that compact bone is made up of what? Remember, bone is a connective tissue, and connective tissues are made up of 2 things. What are the 2 things connective tissues are made up of? Well, I'll give you the easy one. Cells. All right, all
Fibers.
What? I'm sorry. What's the other part?
Fibers.
Okay, fibers and ground substance, and that fibers and ground substance together. What did we call that? There we go.
Inorganic
Matrix. Absolutely. Right in that matrix. The matrix of our bone connective tissue is primarily calcium salts. And so in that matrix of the bone connective tissue is where we are storing the calcium and the phosphorus and things like that. Now, here is one of the important caveats that we have to talk about this. Normally, when we talk about an organ, we want to maintain the integrity of that organ and that it is vitally important. However, as we also mentioned, calcium is vitally important for the function of our cells. If we want neurons to be able to communicate, we need calcium. Muscles to contract, we need calcium. Glands to release their secretions, we need calcium. I know this isn't an issue for any of you because I know all of you had a nice big well-balanced breakfast this morning, which included a big wedge of cheese and a large glass of milk. So you were getting plenty of the calcium necessary from that your body needs from your diet. But believe it or not, there's some individuals that'll have a donut and a Diet Coke for breakfast. And if you had a donut and a Diet Coke for breakfast, are you getting all the calcium that your body needs from the food that you're ingesting?
No.
No. So where's your body going to go to get the calcium that it needs?
The bones.
The bones. Notice if it comes to a decision between having the right amount of calcium and damaging the bones, which is going to win? Do we want to maintain the bone, or is it? Yeah, it's absolutely. So in this case, we may actually have to damage our bones to get the calcium that we need, right? It's because of that. So absolutely. So there in the matrix, we have that calcium, which is obviously important for maintaining bones, but also is important for maintaining, let's say, interstitial fluid levels. Let's just say maintaining calcium levels. So, and these two are not always going to be in sync with each other. Sometimes they're going to be antagonistic. Now, last one. We mentioned red bone marrow. What's the other type of bone marrow?
White bone marrow?
It's not white. It is light in color, but not quite white.
Yellow.
Yellow bone marrow, absolutely. And what makes yellow bone marrow yellow? Anyone know?
The fat.
Fat, exactly, absolutely. Adipose, right? So another big important function of the skeletal system is to store all of that fat, to store triglycerides in that yellow bone marrow. Why might we need yellow bone marrow? Absolutely, right? Triglycerides store energy. Remember, we just mentioned how our bones are very dynamic. So having that fuel source next to it is very, very important. So definitely useful for energy that way. But the other thing, and in fairness, I know we're a far way away from January, so it's hard to remember these things sometimes. But right around January, when everybody's worried about their New Year's resolutions and losing weight and all that, you see the commercials all the time where they show you a pound of muscle and a pound of fat. And which one's bigger?
Fat.
Fat, right? Fat is a very, very light tissue. So the other advantage of putting that fat on the inside, notice you're not wrapping around the outside. It's not providing protection on the outside. We're putting that fat on the inside of the bone, in the center of the bone. And that really light tissue helps to lighten the skeleton. If you think about it, if the skeleton was, if the bones were completely solid calcium matrix, those calcium crystals would be very heavy. So by taking some of these larger bones and putting triglycerides on the inside, not only, as Augustina pointed out, do we have the energy in there for all the dynamic activity for these bones, but it's going to make the bones a lot lighter, making our skeleton easier to move around. Excellent. So there you go. Those are, in fact, the 6 functions of our skeletal system, right? The 3 that we know, the 3 that we talked about before, but then also these other dynamic processes that are going on as well. All right. Questions on that? All right. Excellent. So, much like we did with skin, we want to talk about the cells that are associated with our bone connective tissue. After all, bones are primarily made of bone connective tissue. And so as a connective tissue, as we just mentioned, it is made of cells and matrix. So we need to talk about the cells and we need to talk about the matrix. And let's talk about the cells of our bone connective tissue first. There are 4 main types of cells in our bone connective tissue. Right. And remind me, let's do this up here so that we don't confuse ourselves. They all start from where? They all start from a pluripotent stem cell. And what's the pluripotent stem cell that is responsible for producing all of the cells associated with connective tissues?
That's a mesenchymal cell.
Exactly. Excellent. So one of the things a mesenchymal stem cell can do when it divides is produce an osteogenic cell. These osteogenic cells are unipotent stem cells. which means what again?
They make only one other
Yeah.
cell.
When they can divide to become only one thing. Absolutely, excellent. And one of the other things that's special about these osteogenic cells are these are the only bone cells that are capable of division. Right, we've talked about liver cells and eyeball cells, how they go through that cell cycle, and then they had that ability to undergo interphase and undergo cell division and be able to replace cells that way. But all the other bone cells basically hit that G0 state and they're no longer capable of dividing. So these are the only bone cells that are capable of dividing. And of course, when they divide, they produce one thing and one thing only. They produce an immature bone cell. And what would we call an immature bone cell?
Osteoblast.
Exactly. Excellent. Now, there's one other important thing we need to talk about in relationship to a bone. Let's take, for instance, your humerus and cut it in half, transverse section through it. When we would look at that bone, what we would see is, again, a nice big circle. That would be that bone of the humerus. And as we kind of already hinted at, we know it's going to be hollow in the center. This is an organization I believe we've talked about already in this class. Yes, we talked about it in the hair. We had this candy-coated shell and a chewy nougat center. We've seen this. What do we call the central region again? Like in the hair, the medulla. Excellent. And in fact, this one is called the medullary cavity that we have here in the center of it. Oops. So let's make this a teeny bit bigger. Okay, excellent. This is where we find most of that bone connective tissue. And we'll talk about the organization of that before, although we already know that there are going to be those circular organizing structures. What was the organizing structure of this compact bone again? We learned it in the last section. We have those concentric circles. Excellent, that osteon with the central canal at the center of them. Perfect. But there is something else actually that I want to talk about. On our bone, on the outer surface, is a layer of dense irregular connective tissue that lines the outer surface of our bone, going around the circumference. I'm going to cheat and pull it up a little bit there. This is a dense irregular connective tissue. That happens to wrap around the circumference, the outer surface of our bone. Didn't cartilage have something similar? A dense irregular connective tissue that wrapped around the cartilage?
The perichondrium?
Perichondrium, absolutely. And notice it was a dense irregular connective tissue, but we gave it a name based on its location. The fact that it wrapped around cartilage, we called it a perichondrium. But notice this one wraps around bone. So what do you think we call it?
Periosteum.
There you go, periosteum. All right. It's wrapping around the outside of the bone. The only difference between this and a perichondrium is its location. This one's on bone, so we call it a periosteum. If we took this exact same dense irregular connective tissue and wrapped around cartilage, it would be a perichondrium. And Whoops, nope, want that to stay blue. Turns out on the inside of the bone is another dense, irregular connective tissue that I'm purposely going to move away from the edge a little bit to give me some drawing space. This is also a dense, irregular connective tissue. And since I already wrote it out once, I'm just going to abbreviate it here. That is located on the inside, within the bone. So based on its location, we can't call it a periosteum because it's on the inside. So what do you think we would call something located within the bone, lining the medullary cavity?
Endosteum?
Yeah, exactly. An endosteum. I mention these things right now because these osteogenic cells are located under the periosteum and the endosteum. So nestled here, we'll just draw them as little black circles. Underneath the endosteum, underneath the periosteum is where we would find these osteogenic cells. Excellent. And as we know, osteogenic cells can do one thing and one thing only. And the one thing only they can do is divide to produce our second type of cell. And our second type of cell is an osteoblast. Now, blast are the immature cells. And as we know, immature cells do what?
Make
The matrix.
They make the matrix, absolutely. And the way I remember this in my brain is these are the blasts, and blasts build. B for blast, B for build. Basically, what an osteoblast does is it takes the calcium and the other minerals and components out of the interstitial fluid and basically it deposits it onto collagen fibers. Has anyone ever made rock candy before? You basically take a water, you boil that water, and you dissolve as much sugar as you can into it. You super saturate it because when it's at a warm temperature, you can get more sugar to dissolve into it. Then once you get all the sugar dissolved into it, you lower it back to room temperature where it's now oversaturated, super saturated. And then you take a string that has, and you dip it in some sugar crystals and you put that string in there. And what happens is those crystals on the string encourage those supersaturated sugar crystals in the water to come out of solution and attach. And they basically form these crystals on the fiber of the string. And that's essentially what our osteoblasts does. It takes the calcium and it forms these crystals on our collagen fibers. So it makes matrix. And so that's what our cell is going to do. Let's cheat and put another cell here. We'll make this one a red cell to indicate it's an osteoblast. And it is going to start to make matrix, depositing all this matrix around itself. And as it continues to make matrix and surround itself by matrix, eventually it completely surrounds itself with matrix. And when it completely surrounds itself with matrix, it's basically now in a hole. And what would we call that hole?
Lacuna.
Lacuna. Again, as long as you spell it correctly, I don't care how you say it, but lacuna, absolutely. And now that it's surrounded by matrix, it really can't make any more new matrix, but it can maintain the matrix that is surrounding it. So what would the cell become?
Osteoblast.
An osteocyte, exactly. So basically these cells make their matrix, they surround themselves in matrix. And when that occurs, they basically mature into osteocytes. Which is our third type of cell. Notice I said there are 4 types of cells, but if you think about it, these first 3 types are really just the same cell at different stages of its life. First as a unipotent stem cell, then as an immature cell, and then as a mature cell. And in fact, it really is kind of based on its location, this osteoblast and this osteocyte state. If you were to break this bone and that osteocyte came out of its lacuna, it would basically convert back into an osteoblast, helping to make more bone to repair that tissue and surround itself and mature into an osteocyte again. So really the difference between a blast and a cyte is really its location. If it's not surrounded by matrix and can make more matrix, it's a blast. Once it surrounds itself with matrix, it matures into a cyte. Now, there is one other big difference between the two. When it matures into an osteocyte, the cell changes its shape. The cell becomes very narrow with long processes that come out of it. That's how I spell processes. I'm going to exaggerate the size of this over here. So we've got this elongated cell that has these processes that come out of it. We'll actually see when we look at our lacunas that our lacunas have all these lines radiating out of them. The advantage of this is it allows this cell to communicate with other osteocytes. So maybe there's another osteocyte right here that may be closer to a blood vessel. So it's easier for it to get oxygen and easier for it to get nutrients. Well, what'll happen is its processes will come out and meet with the other's processes, and they will actually form gap junctions. And by forming gap junctions, they're able to share resources. They're more easy to move wastes to maintain these cells. So it allows them, because again, they're stuck inside of this solid calcium matrix, which has moisture, has water in it, but it still can be hard to diffuse the materials that it needs. So by forming gap junctions with its neighbors, it can more easily pass on nutrients, pass on wastes, and these cells can be maintained this way. Now, as I mentioned, our first 3 cells are really the same cell at different stages of their lives. The 4th cell is another completely different cell all of its own, and that is an osteoclast. Osteoclasts actually have more in common with white blood cells than they do with the osteoblasts and osteocytes. Osteoclasts also undergo mitosis, but don't undergo cytokinesis. All right, someone deconstruct that for me and in a common term, explain to me what I just said.
They don't divide. So
They don't divide, but
they replicate, but they don't divide.
excellent. The nucleus divides, but the cell doesn't. Absolutely. Excellent, excellent. So what these cells are, these are actually single cells that have multiple nuclei. What's the advantage of having a lot of nuclei? Well, what does a nucleus contain?
DNA.
DNA. And what's DNA the instructions for?
Function.
Making proteins, absolutely, for making proteins. You're right, for the function of the cell, but to make proteins. These osteoclasts produce a massive amount of digestive enzymes. And their job is to break down the bone matrix. And in fact, that's how I remember them. C for clast, C for chew. Their job is to chew up the bone matrix. And of course, if you're breaking down the bone matrix, you release the minerals, in particular, calcium, because that's what we're caring about here, into the interstitial fluid. And of course, when it gets into the interstitial fluid, where does it go from there? What is intimately associated with our interstitial fluid?
Bone marrow?
Close. Blood, right? Remember, what's in the interstitial fluid, the capillaries exchange materials between the blood plasma and the interstitial fluid. So anything that's in the interstitial fluid, it got from the blood. Anything in the interstitial fluid can go into the blood. So the blood and interstitial fluid interchange. And so from the interstitial fluid, obviously, it can get into the blood. So remember, if you have that donut and diet soda for breakfast, your osteoclasts are going to have to break up some of your bone matrix to release it into the blood to maintain appropriate blood calcium levels so that your body will function normally. Now, most, and of course the keyword there is most, because obviously that means not all, but most of these osteoclasts are located under the endosteum. So they tend to hang out more here on the inside under the endosteum with their multiple nuclei. And notice I drew one other aspect to them. They have this ruffled or scalloped border to them. And that ruffled border basically increases its surface area, making it easier to be able to break down our bone matrix. All right, as you can see here, I've clearly done a truly amazing job of drawing this with my fantastic drawing skills. But let's look at the pretty pictures from your textbook. Notice here we see that cross section from the bone that we were talking about before. Our first cells, remember, are the osteogenic cells. Oops, this should say they come from mesenchymal stem cells. Remember, our mesenchymal stem cells are pluripotent. whereas these osteogenic cells are unipotent. These osteogenic cells are the only bone cells that are capable of division. And like we said, they're found both underneath the periosteum and underneath the endosteum. And being unipotent stem cells, when they divide, there is one thing and only one thing they can become, and that is osteoblasts. B for blast, B for build. Our osteoblasts are the ones that make the bone matrix. Osteogenesis, genesis, give birth to, osteo, bone. They're the ones that make the matrix. And again, they do this by taking that calcium and depositing it on collagen fibers. So they initiate the calcification. They build the bone. B for blast, B for build. Oops. Until they can't build anymore. Once they surround themselves with matrix, then they mature into our osteocytes. Notice, and there's this picture from your textbook where we have this, the processes that come out. I actually like this picture from a different textbook because I think it really shows that nice narrow cell that's located in that nice narrow lacuna that we've seen. And it has these long processes that allow it to form gap junctions with the cells that surround it, allowing them to stay alive and communicate with each other, to share resources. And that's their job, to maintain the tissue, maintain that bone matrix. So again, our first three cells, unipotent stem cell, immature cell, mature cell, are really the same cell at different stages of its life. But the fourth cell of our bone tissue is a completely different animal. Big, large cell with multiple nuclei, known as an osteoclast. C for clast, C for chew. It is filled with massive numbers of digestive enzymes. It has this big ruffled border with this large surface area so that it is able to break down and gobble up the matrix, releasing that calcium and those other minerals into the interstitial fluid, which is going to release it into the blood. This process of breaking down the bone matrix and releasing the calcium into the blood is a process we call resorption. Because remember, as we mentioned, as important it is to have strong, healthy, firm bones, if it comes down to maintaining the bones and maintaining calcium in your blood, calcium is going to win every time. So if we have to break down that bone matrix to get the right amount of calcium in the blood, that is exactly what we're going to do. All right. Questions on that? Sort of. So I like where you're going with this. Absolutely. So for instance, if you are starving yourself, right? Again, summer's over, but at the beginning of summer, you were on the popcorn diet where you were eating one piece of popcorn for breakfast, one piece of popcorn for lunch, and a very reasonable 3 pieces of popcorn for dinner. Obviously, you are malnourished as a result of that. And if you're starving yourself that way, you've got the right idea. Your body does go into protective mode. And fat, adipose tissue, is a very valuable resource. So you are right. It is much more likely to want to hold on to that and it will break down our much more easily digestible muscle to break down to get the resources that we need. So yes, it is. You are correct in that we will break down muscle to get proteins, to get those amino acids if necessary from it that way. But we do the same thing with our bone matrix. So it is similar in that fashion, yes. If you're not getting the calcium you need from outside, if you're not getting the amino acids you need from outside, then we'll break down the stuff inside our body to get what we need. So yes, that is correct. All right. Great question. Any others? All right, so those are the cells of our bone, but we need to talk about the matrix as well. And this is one of the times where I really missed. This whole section is the best section. It's everybody's favorite section because we get to sit in the classroom and we get to hold the bones in our hand and we get to have a lot of fun with it. And this is another example. We have some great bones that have been processed so that we can really see the matrix, because there's basically two main parts to the matrix of our bone connective tissue. Even though when we think of that bone, we think of that skeleton, we think of it as being dried up. In dynamic living tissue, it's about 25% water. It also is about 25% collagen fibers. Remember, as we talked about, collagen fibers are what give it its flexibility, what give it its give, so that when you jump up and down or you jump off the bed onto the floor, your bones have the ability to give a little flexibility, to have a little bit of a give, right? Obviously, there's 50% left, and that 50% is going to be the mineral matrix that we'll talk about in a second. But that mineral matrix in your bone is the same mineral matrix that is on the enamel of your teeth, that is forming the enamel of your teeth. The big difference between your bones and the enamel of your teeth is the enamel of your teeth don't have that collagen. So I think we talked about this before. They don't have flexibility. They don't have give, right? If you're depressed because your New England Patriots couldn't cheat their way into another win this weekend, and Tom Brady's coming back this week to rub it in their faces, so you're drowning your sorrows in a big bowl of clam chowder. And as you bite onto that clam shell, does the enamel of your teeth have any flexibility or have any give? No. And so while it's very hard, it makes it more brittle. So collagen gives it flexibility. We actually have some bones that we have had sitting in acid for months. And as a result of that, all of the bone matrix, the calcium crystals have dissolved away. And all that's left are these collagen fibers. And so we have bones that are basically like rubber bones that you're able to just flex and bend and twist, because all that's left is that collagen fibers. Conversely, we have other bones that we have put into the oven and baked for 20 minutes. This is actually something you can do yourself. The next time you have a chicken for dinner, take the legs, the two leg bones, one of them, put them in a jar filled with vinegar. Don't use acid, but put it in vinegar and leave it in there for about a month. The other one, bake in the oven at about 350 for about 30 minutes, 35 minutes. And what'll happen is at that temperature, it'll evaporate all the water. It will break down all of the collagen. And what you'll be left with is basically this mineralized, hard, rigid as a bone. It'll feel like a rock bone, because you'll have basically evaporated or burned off all of the collagen and all of the water. And all you're left with is that crystalline salt. That crystalline salt is called a hydroxyapatite crystal. Again, one of those alphabet soup words that I guarantee with 100% certainty you will have to spell on the exam. It is primarily made up of calcium phosphate and calcium carbonate, but like I said, there's some manganese, there's some other minerals that are in there as well. So it's these two components, the hydroxyapatite crystal and that collagen, along with water, that make up our bone. And remember also, as we talked about, we're able to make these hydroxyapatite crystals because we are able to deposit them on the collagen fibers. So not only is that collagen vital to give bone its flexibility and give, without those collagen fibers, we can't make hydroxyapatite crystals. There's nothing to deposit them on. So like I said, I don't have any of these things that I can show you, but it is something that you could do now and enjoy by Christmas. So this process of making bone is what we call calcification or mineralization. Again, these osteoblasts take these calcium crystals, the calcium phosphate, the calcium carbonate, the manganese, and all this, and deposit it on those collagen fibers, making those layers of matrix. And when they do this, as we talked about in the last class, they form two different types of bone. All right. We are going to get big into the vocabulary now. So even though it's a little bit early, I think this is a good place for our first break. So any questions before we take our first break? All right, excellent. So let's go ahead and take our first break. We will restart at 9:06. And at 9:06, we will pick up from there. I'll start the recording. Get ready for a vocabulary flurry when we come back. All right, see you guys in 15 minutes. All righty. So as we left off last time, we've now talked about the components of bone connective tissue and how we talked about how the process of making it a little bit, and then how it's used to form two types of bone. We mentioned this in the last section. When we were talking about our connective tissues, we mentioned how bone connective tissue has two types. And the one we focused on for the last exam was on compact bone. Remember, one of the key characteristics of compact bone is it has an organizing structure of the osteons. This is the part of the bone where, like I said, if you were to cut a ribeye and see that nice big circle of bone, what we think of as that smooth, uniform white bone material, as we look at it under the microscope, has many of these osteons, those concentric circles of matrix that are its organizing structure. The other type of bone, which we didn't talk about, we mentioned it in the last class, but we didn't talk about it, is our cancellous or spongy bone. It also has an organizing structure known as a trabecula. Trabecula being the singular, trabeculae with the E being the plural. Again, you get one letter, so you could spell it either way and it would be fine. But these are basically irregular points and processes. Notice this cancellous bone is also called spongy bone because if you grab your kitchen sponge and look at it, with your kitchen sponge, you have all this irregular points and processes. It's not like there's a repetitive pattern on that sponge that you see. There isn't one shape that you could say, this is the shape of what a sponge looks like. It's all of these irregular points and processes. And that's basically what a trabecula is. An osteon is basically a much more uniform structure, whereas trabecula is a much more irregular structure. Now, while there are two types of bone connective tissue in our bones, the organs, the vast majority, about 80% of the bone is compact bone, and that is what you see from the outer surface. All of the spongy bone is found on the inside of the bones. So when you look at a skeleton, you do not see any spongy bone on that skeleton. All you're seeing is the compact bone, and the spongy bone is inside. Now, obviously, there is a lot of vocabulary that goes along with this. Notice here we have a section of a bone that we can see here. They've cut out this section, and we can see here. And notice there are some things we've already talked about that you can identify. Notice here on the outer surface is that periosteum lining the outer surface of the bone. Here we see those organizing structures that are the compact bones, organizing structures that are the osteons. And notice lining that medullary cavity that we mentioned. The medullary cavity is that space on the inside of the bone. Notice there is a thin layer of spongy bone. And again, notice with the spongy bone, there is not just one shape structure. There's all these irregular points and processes, branching processes. Some are thin, some are thick, some are short, some are long. All of these collectively, these organizing structures are known as trabecula. So this is kind of how we see the organization of this. And we have this nice illustration, and we'll go through a lot of the anatomy of this. However, this is a good point to remind you about some of the resources that are available for you. For instance, on our module in our website, under the lab handouts, not only do we have that 30-point skeletal review, your histology that you're responsible for, the bones and bone features we're going to do today, but there are some pictures of some of the materials that are in the classroom. One of them is our bone model picture. This is a model that is in the classroom that basically shows a cross-section through a piece of the bone. And I guarantee with 100% certainty that pictures of this model will be on the exam. Notice, for instance, as we look at this one here, again, we see this tissue on the outer surface. What would this tissue on the outer surface be again? I did turn my microphone on, didn't I?
Dense
Go ahead.
irregular connective tissue.
What kind?
Dense irregular.
Dense irregular, excellent. And how would you identify it based on its location?
Periosteum.
Periosteum. Notice you can't see it quite as well, but you can see a little bit on this view from this here. There's some grayness on the inside of the bone as well. Now, I wouldn't use this on the exam, but since we're talking about it, let's do that. What tissue type do you think this is on the inside?
Loose.
Periosteum.
Well, if this out here was dense irregular, what's this over here?
Dense irregular.
Dense irregular. Here on the outside, we called it a periosteum.
Oh,
What would we call it here?
Endosteum.
The endosteum, absolutely. Now notice if we take a different view from the end, notice this is the inside. We see those irregular points and processes that are the trabeculae, but in particular, we again see the purple here, and this purple that we're seeing here is indeed a much better view of that endosteum. Notice also from this view, we can again see the organizing structures. Notice they've been nice enough to pull some of the layers of matrix that form our osteon out here. So we can see the osteums in the compact bone. And notice they've peeled away some of our endosteum so we can actually see the trabeculae of our spongy bone as well. So these are the organizing structures. They are the trabeculae and the osteon. The osteon is the organizing structure of our compact bone. Trabecula is the organizing structure of our spongy bone. Great view of just the periosteum by itself, but this is the view I want now. All right, so basically, all of this right here is our compact bone. All right. And it is made up of layers of matrix. So if you look closely, for instance, as we see, this osteon is made up by a layer of matrix, but not just one layer of matrix, but many layers of matrix that are basically forming these concentric circles. These concentric circles basically wrap around our compact, our central canal, and they form an osteon. Well, we didn't talk about this before, but we have a name for this because after all, we have names for everything. A layer of matrix is known as a lamella. Lamella being the singular, lamellae being the plural. These particular ones, the ones that I've drawn in dark green, are the ones that are forming the osteons. So as we look at our bone connective tissue, there are three different types. of lamella. The first, the ones I've drawn here in green, are the ones that form the osteons. And if you notice, they kind of form concentric circles around the central canal. So what do you think we call these concentric circles, layers of matrix that form our osteons? If only I had my histology list in front of me, I might be able to figure something like that out. What do you think we call the concentric layers of matrix that form the osteon? What do you think we call the concentric layers? There we go, concentric lamellae. Excellent. Excellent. So our concentric lamellae are one of the types of lamellae, and these are the ones that form the osteons. However, look at the outer surface. When we look at the outer surface of our compact bone, notice here the layers of matrix are much straighter. Right? They've even raised some pieces up so we can see how flat they are. Notice, in fact, these layers look like they might go around the entire circumference of the bone. They're so straight. What do you think we call these outer layers of matrix that would go around the entire circumference of our bone? There we go. They're circumferential lamellae. They go around the outer surface of the compact bone. Or we could think of it as these are the layers directly under the periosteum. So we have circumferential lamellae, we have our concentric lamellae, but if you look closely, you can see that, for instance, right here are some layers of matrix that aren't really forming a circle, but aren't on the outer surface. Here's another little patch of matrix filling up that space. Here's this kind of more weird shape. Notice these layers of matrix don't have a distinct shape to them. Instead, they're kind of filling up the space inside the tissue. And what do you think we call these layers, these irregular shaped layers of matrix? That fill up the space in our compact bone. Interstitial, excellent. So notice, as I mentioned, we have three different types of layers of matrix: concentric, circumferential, and interstitial. And notice one other important thing about our compact bone. When we're talking about our compact bone, not only can we define it by its organizing structure, which is the osteon, but notice compact bone contains all three types of lamellae. So our compact bone is going to have concentric, circumferential, and interstitial lamellae within them. Let's change our views. Let's go back to the edge. Notice on the inside where we have the compact bone, pardon me, where we have the spongy bone, right? It's lining the medullary cavity. Remember, its organizing structure are the trabeculae. And clearly it has to be made of layers of matrix. However, these layers of matrix, here's a long one going down along this way. Here's a relatively short one. Here's kind of a narrow one. Here's one that's kind of branching. Notice here when we have these layers of matrix, are any of them going around the entire circumference of the bone? No. Are any of them forming nice circles around a central structure like a central canal? Are any of them forming osteons?
No.
No. So notice with our spongy bone is only made by interstitial lamellae. But if you think about it, it makes sense. Trabeculae have irregular points and processes. They have no uniform shape. And interstitial lamellae have no uniform shape. So our spongy bone is only made by those irregular lamellae forming irregular trabeculae. So when we look at our matrix, we can see this. Let's go back to the pretty picture. So again, notice here is one of our osteons. We pull that osteon out and we see the concentric lamellae that are forming them. Notice they surround a central canal, which, as we talked about, contains the blood vessels and nerves. Notice one more thing. Notice that, again, here they've illustrated for us the collagen fibers that you would see in these lamellae. And notice the layers, the collagen fibers go in opposite directions with each concentric lamellae. This again is what gives it that resistance, that flexibility, that give to that bone by having those collagen fibers running in different directions. Here's a picture we've seen before. Here we're seeing an osteon, that organizing structure. We have already talked about our central canal that is at the center of that. Haversian canal is a much, much older term. A good old Bob Haversian was the first one who described this, and they named it after him. So that's something, again, that you may see in some of the textbooks, but is not a term you will hear me use ever again. And we can see those lacunas that we talked about. But notice now that we're looking at this nice high resolution view, not only can we see the lacunas, but notice we can see these black crack-like structures that are coming out of them, radiating out of them. These are also spaces because remember, as we know, our osteocytes, the mature cells that are in these lacunas, they have long, thin processes that come out of them, where again, as we talked about, they form a gap junction so that one that's here closer to the blood vessel can get that blood and oxygen, nutrients, and send it to the other cells, and these cells can send their waste towards the blood vessels. So notice these little tiny spaces that radiate out of our lacunas are like tiny little canals, and in fact, that's what they're called, canaliculi. Canaliculi are tiny little canals. So we have that large lacuna with the tiny little canaliculi as the spaces that are radiating off of them. Notice our illustration here, not our illustration, our light microscopy here does a nice job of showing this. And notice if we go back to our model, notice here on our model, they've shown us this with a little bit of a lacuna. And then we have those canaliculi radiating out of them. If we look at it from the top view again, or is that top view? Excellent. Notice here we can see the lacunas. So there's a lacuna, there's a lacuna, there's a lacuna, there's a lacuna. But notice on this one here, they've put a little bit of the processes coming off of the osteocytes. for those canaliculi. So we can see how they've done that for us there as well. So our model maker's done a nice job of showing us that as well. Here's the illustration emphasizing that point we were talking about before. Concentric lamellae form the osteons around the central canal. Circumferential are on the outer surface and go around the entire circumference of the bone. And interstitial lamellae fill the spaces in between. They're the irregular layers, shape layers that fill the spaces in between. Again, notice here in the center, our spongy bone is only going to be made up of interstitial lamellae. But I want you to notice something else as well. Notice all our osteons basically run parallel and run the entire length of the bone. Now, that doesn't mean that when you have a long bone, you have blood vessels that enter at one side and exit the other. We need a way to get blood into and out of. And so notice out here in our periosteum, we have some blood vessels that would be hanging out here in the periosteum. And notice that their blood vessels would then penetrate into and connect the blood vessels that are in our central canals. And so, of course, we have to give fancy names to these. As we know, the space that contains the blood vessels that run parallel at the center of the osteons are our central canals. But the ones that penetrate into the bone between the central canals are known as the perforating canals. Now, you may also hear the term Volkmann. Again, that is not a term I will use, but you'll see that. And notice also, if we look at our model picture, notice on this side, we can see those blood vessels coming in and going up through the central canals. But what's cool is if we look at the other side of the model, they've conveniently enough removed those blood vessels for us, so we can actually see the spaces. So again, this space, this space, this space, all of these spaces that are running parallel to the osteons, those are our central canals. And all the ones that are going across them and connecting them, hold on, let me just finish what I was saying, would be the central canal. Yes. What's your question?
So like on the lab exam, how would you ask these questions? Like, would you point to it and be like, what is this canal? Or would you be, what is the space?
Space. All of these things are spaces.
Okay.
Yep.
Okay.
Now let's look at one other thing. Okay, let's do this. I haven't looked at this yet, so I don't know how successful this will be. But another resource that you definitely, absolutely, positively will be using in this section, if you haven't started using it already, is your practice anatomy lab. We'll talk about the bones that you can see here, but I was curious about the histology. Let's see if under connective tissue, we have gallery, bone. Perfect, that's what I want. So let's get rid of the labels here for a second. Nope, don't want the labels. So notice as we look at this, we can clearly tell that this is compact bone because we can see the osteons. And this space here indeed is the central canal. We comfortable with that? Notice at this lower magnification, we really can't see the canaliculi. We know they should be radiating out of those spaces, but we can't see it. But what we can see is, notice there is a central canal here and a central canal here. And then there's this space in between them. Notice there's a little space here coming off. Any idea what these spaces I'm coloring blue might be?
Spongy bone?
Not a bad guess, but remember it's a space.
Oh,
Yeah.
Arteries and veins would fill the space, but again, I'm asking you to identify the space. Let's think about this again. Notice this over here is a central canal, This over here is a central canal. And what is the space that connects central canals? There we go.
Perforating canals.
Mitch has got it. Perforating canals. Notice even under the microscope, we can see a perforating canal. Basically, what's happened right here is if you go back to and think about this picture here, basically they have made a section through the bone that is kind of going right through there. So we can actually see that lateral space. So that's what you can see. So histologically, we can see these as well. So to answer your original question on the exam, I could basically have this and ask 3 questions. Well, I can ask many questions, but 3 space questions. Identify the space, or identify the space. All right, what's this first space right here?
Central canal.
Central canal,
Second space right here.
perforating canal,
Third space right there.
Lacuna.
Lacuna, exactly. Now I
For
didn't,
okay,
go ahead.
sorry. So for the interstitial lamella, how would you
Layers.
layers?
So
Okay.
let's go back to the illustration because it's even simpler there. Right, on the exam, identify the specific layer.
Concentric.
Lamella. Identify the specific layer.
Interstitial.
No, this one.
Sorry.
Circumferential lamella. Identify the specific layer.
Interstitial lamella.
Yep. And again, remember, compact bone has all of those. And then even easier, identify the specific layer. Yep, there you go. Sarah's got it. Interstitial. Remember, spongy bone is only made of interstitial lamella. And we'll take a closer look at it in just a second. All right. Ava, did that answer your question?
Yeah, thank you.
Perfect. All right, we did that. Perfect, excellent. So notice in the lining the medullary cavity and also within the heads of our bones, of our long bones, is where we find our spongy bone. And notice now when we look at it, you can see why it gets the name, because it really does look like those irregular points and processes of a sponge with no uniform set shape. And so again, those irregular organizing structures are called trabeculae. And notice what they've done for us nicely here is taken a section through a trabecula and peeled off the endosteum. And we can see it is also made up of layers of matrix. It has lacunas, it has canaliculi. It has all those things we expect to see in bone, but notice it's not going around the entire circumference of the bone. So these are not circumferential lamellae. They're not around a central canal. So they're not concentric lamellae. They're forming irregular points and processes. These are only interstitial lamellae. Spongy bone is only made of interstitial lamellae. And notice they've shown us the osteogenic cells, the immature cells, and they've even given us an osteoclast. So we can see that on this nice illustration as well. And again, if you think back to the picture, notice as we go to the end view, again, it's not the best view of it, but you can see the lines in here forming the layers of matrix. And again, there's no uniform shape to them. So all of these trabeculae that we can see are made out of made up of interstitial lamellae. All right. Questions on that? So notice some of this was material we already knew. We already knew about osteons. We already knew about central canal, but we have added more vocabulary. Oh, I just thought of one more thing that we did not mention. Hopefully someone has our histology list to see if I've forgotten anything else. But Let's do the top-down view. I like that the best. Notice, as we've talked about, out here we have our periosteum on the outer surface. As we know, it houses the immature cells like the osteogenic cells and the osteoblasts. It also stabilizes blood vessels, anchors blood vessels, and holds them in place. But this periosteum does something else. It protects the bone, and it's an attachment points for ligaments and tendons. Remember, one of the things we talked about, that ligament can be so strong that when you pull on it, you're more likely to break the bone than you are to tear the ligament. One of the reasons it has so much strength, and you can see that as you look at it here, is the collagen fibers that go into it actually penetrate deep into the bone matrix. So these collagen fibers coming off of it penetrate deep into the bone matrix, helping to hold it and anchor it in place. These extensions of collagen fibers are what are known as perforating fibers, but they have another name as well. Anybody have the histology list in front of them might know what the other name for these are?
Sharpie.
Sharpies.
Sharpie fibers, exactly, because good old Bob Sharpie was the first person who identified them. So they're called the Sharpie fibers after him. So there you go. So we see those as well. Notice we don't have perforating or Sharpie fibers on the inside, because on the inside of the bone, we don't need that protection. Ligaments can attach to the inside of the bone. We still need to house blood vessels. We still need to house immature cells. So the endosteum does that, but it's not providing that attachment point. It's not providing that protection because it's on the inside.
How would we label the yellow bone marrow? Like,
So the great question. What I would say is that this picture doesn't show yellow bone marrow per se, but we know there is a space here. This space here would be the medullary cavity. And in a mature bone, what would be contained within this space?
the yellow bone marrow.
Yellow bone marrow, exactly.
And that's the same for the spongy bone with the red bone marrow.
Exactly.
Okay.
Yep.
And then so for the low and high resolution, we, I guess, look at two different like pick or histology slides
again,
of the compact.
I haven't looked at all of these bone slides, but let's see if there we go. Notice here with a high magnification, we can clearly see the canaliculi. All right. So we can see those nice canaliculi that are coming out of the lacunas. Whereas if we go back to the lower magnification, right here at a lower magnification, we can see, for instance, the perforating canal really nicely. But can we see the canaliculi on this particular magnification? No. So that's the advantage of, so at a low magnification, there's some things that are going to show up a little bit better. And at a higher magnification, there's some other things that are going to show up a little bit better. Right? Notice here, we basically just have the single osteon in here. So we can't see, for instance, the interstitial lamellae, and we certainly can't see the circumferential lamellae. But if we look at a low magnification view, which is essentially what this is, we can see the circumferential lamellae, we can see the periosteum and the endosteum, interstitial lamellae, circumferential lamellae, right? So there are some things we can see better at a low magnification and some things we can see better at a high. Okay. All right, any other questions? I think that is everything on your histology list, right? Have I caught all of it now with the Sharpie fibers?
Yeah.
All right, excellent. So we can now switch from the microscopic anatomy to the gross anatomy. And for the gross anatomy, we need to first talk about our structural classifications of bones. Right? We talked about structural classifications of glands, and that's basically we are identifying something structurally, which means by their shape. Now notice I've got 4 with an X through it. there are truly 6 structural classifications. Your book does emphasize, but your book does identify all 6, but it doesn't fully emphasize them all the same way. It emphasizes the big 4, and then it kind of mentions the other 2 as a side. So I want to make sure we emphasize all of them. So there are indeed 6 structural classifications of bones. And so let's figure out what those are. Give me one of the ones. Can be one of the obvious ones. Give me one of the obvious structural classifications of a bone. What kind of bones do we have?
Long bone?
Long bones. Excellent. How would you define a long bone?
It's long.
It's long, yes. The building is tall because it is tall. What does it mean to be a long bone?
To be long and relative to the body, I guess.
Okay, I kind of like where you're going with that. You've got the right idea, absolutely. A long bone basically is a bone that has more length than width. So it is a long, narrow bone that way. But typically it also has enlarged ends to it. Those heads at the end of it. This is kind of like those Dino bones that you think of when you watch the Flintstones, right? They give Dino his bone, and there's this big, huge bone with the big, huge ends, right? That Dino bone is kind of what we think of in terms of the long bone. We've got this bone with a elongated shaft and 2 enlarged heads at the end of it. What's another structural classification?
Short, flat, and irregular bones.
Sorry, say again?
Short, flat, irregular bones.
Okay, well, you've given them all to me, but perfect. Excellent. Short is one of them, short bones. What do we mean by short bones? They're
Bones
short.
that are short that have a greater width than length.
Okay, in this case, they're small, kind of block-shaped bones. All right, so they're more cuboidal in shape. So they're kind of block-shaped or cuboid-shaped bones. Excellent. I heard you say flat. All right, other than being flat, how would you define a flat-shaped bone without using the word flat?
Not squished, but like thin.
Okay, no, I like that.
And
Thin and wide bones. They're thin, wide bones, but there's like my hand, thin and wide. But there's one other characteristic that all flat bones have as well. Not only are they thin, not only are they wide, but what else do all flat bones have?
are they
Curvature?
broad?
Yeah, they all have a curve in them. Absolutely. So they all have a curve in them. Again, here's our block shape short bone. With our flat bones, right, there are these narrow bones with a curve in them. So broad, narrow, with a curve in them. And then you also mentioned irregular bones. So how do we define irregular bones? Well, this one's easy. An irregular bone is a bone that is not long, short, or flat. So this one we can cheat with our definition. The vertebrae are a great example of that. These are the 4 main classifications that we think of. And why are these the main classifications? Why are these the ones that most people emphasize? Not only are they the most common, and you're absolutely right, they are the most common, but almost all named bones fall into one of these 4 categories. So if you randomly pull a bone out of the body that has a name on it, it's almost certainly going to fall into one of these 4 categories. Of course, what's the most important word in that sentence?
Almost.
Almost, because almost means not all. There are two other categories of bones. The first are what are known as sesamoid bones. Sesamoid bones get their name because, again, most are the size and shape of a sesame seed. These sesamoid bones are found in tendons. They help to change the shape of the, or the angle of the tendon to help to improve the movement. And they are, for the most part, irregular in their location. Some of them are going to be located, their locations are going to be determined genetically. Some people will be more common to receive them in the wrists, whereas other people may be more common to receive them in their ankles. They can be also not just genetically predetermined, but also by function. Bull riders, professional bull riders, for instance, will get this in the tendons on the underside of their thighs from this activity. So again, remember we talked about how there's 206 named bones. Not everybody gets these bones and not everybody gets them in the same place. So if I have a bone right here in my wrist, can I call it Sally and then it's going to be Sally for everybody?
No.
No.
No, because as a result of that, different people have them in different locations, so we can't give them names. So most of these, I see your question, I'll answer in a second. Most are unnamed because people have them in different places. There are a couple exceptions, and for our sesamoid bones, what's the exception? What is the largest and the most common sesamoid bone? There you go, the patella, which absolutely is 100% correct and a very fancy word. Can anyone tell me what the patella is commonly known as?
Kneecap.
Your kneecap. Absolutely, your kneecap. So almost everybody has two. Notice what I said there. There actually are some people who don't develop a kneecap, or in some cases don't develop either kneecaps, but most people have two kneecaps. Now, to answer Sarah's question, a bone spur is typically caused by damage to the bone. Well, okay, there are a couple things that can cause it, but one of the common causes is a bone gets damaged, and as it gets damaged and starts to heal, a piece of bone will start to grow off of that as a result of it. So you get abnormal bone growth typically caused by some type of injury to the bone. And so you get, a bone that has a normal, typical shape starts to get an irregular formation on it. And those can actually be gone in and removed surgically as a result of that. So typically those bone spurs are caused by irregular bone growth, often associated with damage. So not quite the same thing as the sesamoid bones.
Because
Go ahead.
Like for the structural classifications for the 5th and 6th one, are we going to be responsible for that?
Yes,
Histology.
absolutely.
Okay.
Yeah. Well,
Okay.
I mean, not histology. This is gross anatomy that we're talking about, right? So,
I mean, yeah.
right, you need
Sorry.
to know the patella is
Yeah.
a sesamoid bone, right? Long bones, although we didn't mention it, I think you guys kind of hinted at it. All the long bones, for instance, all of them are found in the appendages. So the appendages are where you find all the long bones. However, are all the bones of the appendage long bones? Well, I'm asking the question, so what's the obvious answer? No.
No.
Like on the arm, what would the exception be? Come on, I know you know, even if you don't know, you know.
Your elbow.
No, not a bad guess, but no. Where do you have bones in your arm that are not long bones?
Fingers?
Nope, fingers are actually all long bones. Little lower.
Wrist.
Yeah. What are your wrist bones called?
Carpal.
carpals. The carpals are short bones. Your heel and ankle bones are short bones. So those are the exceptions in the appendages. All right. But notice there's one more structural classification. This structural classification is found only in the skull. The bones of our skull are somewhat interesting in the way they form. Let's cheat. Here's a piece of the skull right here. And in this piece of the skull, when the two bones of the skull knit together, they don't typically come together in a straight line. They typically have this kind of irregular intermeshing where they come together. Now, of course, if this is bone A and this is bone B, as convoluted as this line is, clearly this right here is part of bone A. Clearly this right here is part of bone B. However, what will happen in our skull is as these bones are growing together, in the spaces in between, an additional bone may form. This additional bone that forms is its own little bone. Now, these joints of the skull are known as sutures. So these bones that form in them are sutural bones, or what are also known as wormian bones. And they form, the most are the size and shape of a grain of rice. So they're really, really small, and they only form in sutures. A typical person will have dozens of these. And while most are the size of a grain of rice, some can be larger than a quarter in size. So they can get quite big. However, while every single person has dozens of these, do we all have them in the same locations, between the same sutures, in the same size, in the same shape?
No.
No. So notice every single person has far, far more than 206 bones. Well more than that. But 206 is the number we use because that is the number of named bones we have in the body. So most of the sesamoid bones are not named. None of the wormian bones are named, but we have a ton of them. So they're there as well. And notice I've done an amazing job of drawing these things, but let's look at the pretty pictures from your textbook. Here we see that broad, thin, curved, flat bone, like the one in the skull. There is our long bone with that elongated shaft and the enlarged heads. Here are some of those block-shaped short bones, like in the wrist. Our vertebrae is a great example of an irregular bone. Here we see a nice large sutural bone located between the sutures of the skull. And there's our largest and most common sesamoid bone, the patella. So every bone in your body falls into one of these four categories. But as I mentioned, the vast majority of the bones in your body are going to be long, short, flat, or irregular. Collarbone is an example of a flat bone. All right. Questions on that? How are we doing on time? Excellent, excellent, excellent. All right. We have identified the classifications of the bones, but we need to talk a little bit more about the anatomy. And let's first look at the anatomy of a long bone, identify some of the anatomy. So we have some more anatomy to go over here. So let's bring up a new whiteboard. And like I said, we'll draw a quick, simple, easy Dino-shaped long bone. Again, we are going to have an elongated shaft with two enlarged head-like structures at the ends. We already know some of the anatomy. We know that in the center of the shaft, we have a hollow space. And what is that hollow space called again?
Is it the medullary cavity?
Excellent. We know that medullary cavity is going to be lined with a little bit of spongy bone on the inner surface. And we know that there is going to be a dense irregular connective tissue that lines the inner surface of this medullary cavity. And what would we call this dense irregular connective tissue based on its location?
Periosteum.
Endosteum, excellent.
Sorry.
Again, I don't care how you pronounce these things as long as you spell them correctly. Excellent. We also know we have a dense irregular connective tissue on the outer surface as well. And what did we call that one based on its location?
Periosteum.
Periosteum. Excellent. So those are things that we've already learned and we already know. So we can just put those on there because we're comfortable and we understand that. Now, as I mentioned, this long bone, and we are indeed doing the anatomy of the long bone. I mentioned it has a singular elongated shaft, but are we going to get away with a term like shaft in an anatomy and physiology class?
No.
No, of course not. So what is the appropriate anatomical term for the shaft of the bone?
Diaphysis?
Yep. It is the diaphysis. Excellent. We also have two head-like regions. And what is the appropriate term we're going to use for that head-like region?
Proximal epiphysis and distal epiphysis?
Absolutely. We are going to indeed refer to them as epiphyses. Epiphysis is the singular, epiphyses, ES is the plural. And you've also hit on another important characteristic. We have two epiphyses, so we need a way to tell them apart. And remember, all of our long bones are in our appendages, which means one end is going to be closer to the core of the body, and one end is going to be further from the core of the body. And as you mentioned, the end that is closer to the core of the body, how are we going to refer to that?
Proximal.
Exactly. So we have a proximal epiphysis and we have a distal epiphysis. Excellent. All right. So far, so good. All right, let's start talking about some of the other things that we know. We know, for instance, that on the outer surface of the bone, where it's going to form its joint with the bone next to it, we have a special connective tissue. What connective tissue are we going to have on the ends of the bones that are going to help them to form their joints?
Cartilage.
What kind of cartilage?
Elastic?
Do we want them to be really rubbery and bouncy?
Oh, no,
No,
sorry.
there we go. We want the hyaline on the edge of the bones. Now, of course, this hyaline cartilage at the end of the bone, is this the only place we find hyaline cartilage in the body? No. So we're going to give this cartilage at the end of the bones a special name. We call it articular cartilage, because to articulate means to form a joint. right? So to articulate, form a joint. This is the articular cartilage. This is the cartilage that is used to form a joint. All right, now, one last thing that you guys have mentioned for me already. Remember, as we mentioned in, oops, no, I want that to stay. Oh, we'll make it green. As was mentioned earlier, in an adult like yourselves, what is this medullary cavity going to be filled with?
Adipose.
True, and that adipose is going to turn the marrow into what type of marrow?
A red bone marrow.
Well, if it has adipose,
Wait.
it's not red. What is it?
I mean yellow, sorry.
Yeah, in an adolescent, in a baby, the bone marrow in the medullary cavity is going to be red. But as we mature, we need to store that energy. We need to make the bone lighter. So it's going to be filled with yellow bone marrow.
Is that why when babies' bones break, they heal faster?
Yeah, I think it has more to do with the overall metabolism of the baby and the baby's cells than it does for the adolescent. So I don't think it's necessarily that it's the bone marrow. It's just they need more red blood cells. They're more metabolically active. So things like that. But as adults, we still need to be able to make red blood cells and white blood cells and platelets. So as someone mentioned earlier, and as we saw, remember the inside of the heads don't have a medullary cavity. Inside the heads, we have all this spongy bone, and I know I made that look really regular, which I shouldn't have, so let's just cheat and do it this way here. We have all the irregular points and processes of here of our spongy bone. So we have our spongy bone in the epiphyses of it, and the space inside the spongy bone is where you're going to find the red bone marrow. Anybody here ever donated bone marrow before? Know someone who's donated bone marrow?
Yes.
Yeah, I have.
Oh, wow, you have. Excellent. How did they do that? Where did they get it from you?
I don't remember.
You don't remember?
Like a
Someone
long
else said
needle
yes.
or I don't know.
Someone else said yes. Where did they, anyone know where they go to get the bone marrow? Exactly. They take this huge gauge needle and they want to put it into the bone where there's going to be the most marrow. So in an adult, that's typically either the hip bone or the head of the femur, your leg bone. So they'll take this big, huge bore needle, they'll inject it in there, extract some of the bone marrow. And are they able to get enough from just one injection of that needle to get enough bone marrow for that donation that they need to do? No. 3, 6, 9 times they'll go in there extracting it out. It is an excruciatingly painful process and an incredibly generous gift for someone to be able to give because that's where they have to go. It's not easy to extract from these locations and it can be a very painful process. So they're inside this space. And again, remember too, we'll have endosteum lining these spaces as well. So I'll cheat and draw a little endosteum in there as well to remind us it's there. But the ends of the bones aren't hollow. They're filled with spongy bone, whereas spongy bone just lines the inner surface of the medulla. Now, there's actually one other place where we find cartilage, hyaline cartilage as well. Right? Notice we have this distinct diaphysis and a distinct epiphysis, and in between them is kind of this transition area. This transition area is what we would commonly refer to as the neck of the bone. Of course, we're not going to get away with a term like neck in a class like this. So what is the appropriate term for the neck of the bone?
Metaphysis.
Metaphysis, exactly. And again, I don't care how you pronounce it as long as you spell it correctly. The metaphysis. And again, we're going to have a proximal and a distal metaphysis. So let's go ahead and be correct and make this one distal. Metaphysis. In this metaphysis, in this transition region, we are going to find another chunk of hyaline cartilage. This little bit of hyaline cartilage that is located in the metaphysis is again still hyaline cartilage. But this hyaline cartilage is in a different location and serving a different function, so we give it a different name. Anybody know what we commonly refer to this hyaline cartilage as? I know you know, even if you don't think you know. This chunk of hyaline cartilage in the neck of the bone is what we commonly refer to as the growth plate. What happens in the growth plate?
Growth.
What kind of growth? This is where the bone grows. How?
Does the cartilage extend?
It
Or
does.
grow?
We'll talk about how that happens. This is what causes the bones to grow in length, right? How many of your long bones are the same size now as they were when you were born?
No.
None of you, right?
Hopefully.
Because this is where our bone grows in length. This is where our bone grows longer. And this hyaline cartilage, this growth plate, is what is known as the epiphyseal plate. Excellent. That's how you got from a little tiny baby to the size you are now. Are you still continuing to grow? Anybody here still getting taller?
No.
No, probably not, because most of you have ended puberty at this point. And what happens when we end puberty is that this epiphyseal plate ossifies. Instead of hyaline cartilage, it becomes a small, thin line of compact bone. And when it becomes that small, thin line of compact bone, at that point, we can no longer grow in length. And instead, we basically have an epiphyseal line. And at that point, our bones don't get any longer.
You said after puberty?
Yep. So usually sometime between 18 and 25, our growth plates close. Right? If you just randomly found a bone in your backyard and it happened to be cut in half, you could look at that to look to see if there's cartilage there, if it's an epiphyseal line or epiphyseal plate, and that'd give you an idea of the age of the person that bone belonged to. If it was ossified, then they're typically older than 25. If it's fully cartilage, then they're typically younger than 18. If it's some kind of weird mix of bone and cartilage, then they're probably somewhere between 18 and 25. So the condition of this epiphyseal plate or epiphyseal line can give us some idea of the age of a person just by looking at their bone. And notice, yes, you could take the bone and cut it in half, but the nice thing too is, do you think cartilage and bone look the same under an X-ray?
No.
No. So if we X-rayed the bone, we'd be able to tell. Is it fully cartilage? Is it fully bone? Is it somewhere in between? And it would give us some sense of the general age. Excellent. So notice, we've done a nice job of doing this with my, again, truly amazing illustration, but let's go through with the pretty picture from your textbook. Again, our shaft is the diaphysis. The epiphyses are the heads, and we distinguish them by proximal and distal because they're always in the appendages. So one's always going to be closer to the core of the body, and that will be proximal, and will always be further away from the core of the body. That will be distal. Articular cartilage is the cartilage that forms the joints at the ends of the bones. And what kind of tissue did we say that was again?
Hyaline cartilage.
Hyaline cartilage, excellent. We have that transition area, the neck region known as the metaphysis. And in the metaphysis, we're either going to have, notice like this one has a thin line of compact bone, which tells us that the epiphyseal plate has closed and it is forming the epiphyseal line. Whereas this, if this was an immature bone, then we'd have a thin line of cartilage in this space, and that would be the epiphyseal plate. As we mentioned, we have the periosteum on the outer surface. What type of tissue was it again? Dense, irregular connective tissue. Excellent. Its function, remember, is to act as an attachment point for tendons and ligaments, hold blood vessels in place, provide protection, and house the immature cells. Our medullary cavity contains our yellow bone marrow and is lined with the endosteum that houses our immature cells. And that is our anatomy of the long bone. Questions on that? All right, we have done the anatomy of the long bone. We still have to do the anatomy of the short bone, the flat bone, and the irregular bone. And that makes me tired just thinking about that. But the good news is that the anatomy for all of them is basically the same. So we can lump them all together. With our let's just do this. A short, a flat, or a regular bone, they basically have the same type of anatomy. For starters, they on the outer surface are going to have a thin shell of compact bone. Remember, of course, we have to have compact bone on the outer surface. We never get to see the spongy bone that is showing. So we have that compact bone on the outer surface. And because it's forming the outer surface, what do we use for that term? What's that term we've used for the outer surfaces of things? Medulla is the center, and what is the outer surface called?
Cortex.
Cortex. So we have a thin cortex of compact bone on the outer surface of it. So we've got the cortex there and the cortex here. Of course, that is going to have its periosteum on the outer surface. And then on the inside, we are going to have, oh, no, I want this to be black. All the irregular points and processes that are the trabeculae of our spongy bone. And that's good enough. We'll cheat and just do a small portion of it. As we know, this is made up of all of those irregular shaped interstitial lamellae. So again, the organizing structures are known as trabeculae. And they're made of irregular layers of matrix. And what do we call those irregular layers of matrix again? What do we call the regular layers of matrix that make up our trabeculae? There we go. Interstitial lamellae. Perfect. This space is also going to be aligned by a dense irregular connective tissue, lining these spaces. And of course, this dense irregular connective tissue is our endosteum. And the space itself, even in mature adults, is primarily filled with red bone marrow. And there you go. The anatomy of our short, flat, and irregular bones is really just that simple. Except for one incredibly stupid thing. As we know, anatomists hate us. And one of the ways they show that hate is by making things unnecessarily complicated. This structure that I have pointed at right here, what is this organizing structure again?
Trabeculae.
It is a trabecula. What type of layer of matrix is it made of?
Interstitial lamellae.
Excellent. It's made up of interstitial lamellae. That is absolutely true. However, for some reason, and I have no idea why, in flat bones and in flat bones only, they give this structure a special name. And that special name it gives for the structure is diploe, with the little umlauts on top. And I don't know how to do an E with an umlaut. So let's cheat and do it this way. There. So the trabeculae of a flat bone are referred to as diploe. Why just in a flat bone and not all the others when they have the exact same anatomy? I don't have a good answer for you. But notice if we go to the pretty picture from your textbook, again, we have that cortex, outer compact bone, spongy bone in its core, which again is called diploe in flat bones, and it typically contains red bone marrow. All right. Questions on that?
So if you pointed to the organizing the structure on, let's just say, an exam, would you specify like which, I guess, bone type of structure
Here's,
bone it would be?
let's do that. So on the exam, I have this arrow right here, and I could ask 3 questions. Identify the organizing structure, and your answer would be
Trabecula.
trabecula. Excellent. Identify the identify the specific layer of matrix that forms this structure.
Interstitial lamellae.
Identify this structure in a flat bone.
Diploe. Or
There you go.
diploe. And then do we need to put the 2 like dots on top of it?
Don't worry about that.
Okay. Okay.
Right. Excellent. So yeah, that's how, and so I would have to say that. Identify this structure in a flat bone. What would you call this structure in a flat bone? And that's why we would do that. Okay. All right, great questions. Any others? All right, here's what we are going to do. we're going to take our next break. When we come back from the break, we are going to do our histology first. I want to look at some of these things that we've been talking about under the microscope and talk about that. We've already looked at some of them, but I want to go over it a little bit more, make sure we're comfortable with that, and talk about the histology a little bit more and do that. Look at compact bone, look at spongy bone, do that. Then after that, we are then going to form our groups for the presentations. I would strongly encourage you, if you've not done so already, to either print out or at least on your phone or your iPad or something like that, grab so under the modules, under the lab handouts, bone and bone features. So if you've not done so already, please print out or grab or look at this on your phone or whatever, because this has the bones and the bone features you're going to be responsible for. So I need to talk about these things so we can discuss what the game plan is, and I can give you guys your responsibilities, then break you up in your groups, and you can spend the rest of the group time talking about and planning your presentations. All right. Questions on any of that? Let me do one thing here while I'm thinking about it. There, make that change. Perfect. Okay, excellent. So, oh, I just lost all of that. So if you haven't done so already, the histology list would be useful. So we're going to do histology first. Then we're going to talk about the bones and bone features. And we'll do this after our break. So it is 10:22 right now. So let's take our next break, come back. We will restart at 10, what'll that be, 37? And I'll start the recording at that time. All right, any questions before we take our next break? All right, I'll see you guys in 15 minutes. All right. So again, taking advantage of the resources that you're using. Notice there's not nearly as much histology on this exam as there was on the previous one, but I still want to remind you of the resources that you have available for you. As we finished talking about, as we were showing before, your practice anatomy lab has bone, both high magnification and low magnification views of that. For the compact bone, it also has spongy bone. Notice with the spongy bone, again, we can clearly see these irregular points and processes that are the trabeculi. And if we get a closer view, which hopefully they will give us, with this closer view, we can, oops, there we go. We can kind of see the layers, although there's some better views. Not much better. Come on, I don't want that. There we go. No, we can't really see that well, but some of the stains will show it nicely. Okay, so that doesn't work. But some of the other resources, remember that you have as well. When we go back to our, where's the one that I want? This one. So again, when you go back to your, oops, modules, under your modules, under the study guides, they have the Yale histology. When you click on the Yale histology, this is a great resource for histology. Hopefully you took advantage of it for the last test, but if not, start using it now. When you go to the bones section, as you read through all the information down here, they have some of the information you're responsible for. So for instance, here is our trabecular bone. So when we want to look at that, again, we can see those trabeculi with the lacunas with the osteocytes in them. Notice we can see the red bone marrow and some blood vessels and stuff, and they have some great descriptions. Those are great resources that you can use. The other one that will help you as we are moving forward is to notice there is this bones anatomy standard. And if you go to this website, this is an amazing website with some amazing pictures of some of the, we can grab that and look at different views as we look through this here. But again, for instance, we're going to start learning about the skull. So if you're interested in, for instance, the occipital bone that you are studying, there are some amazing views of this that you have that you can look at, that you're able to adjust and modify. You can download these things. You can see some of the labels and some of the characteristics that are on them. Great views. It's a great way to see that hypoglossal canal, which can be challenging to see sometimes in some of these things. Now, obviously, there's a tremendous amount of detail. Are you responsible for the names, for instance, of all of these grooves and curves and raised ridges on this bone?
No.
No. How would you know what it is that you're responsible for on this particular bone?
It's on our list.
Exactly. Right. The other thing that you have here is this bone and bone feature list. And for that occipital bone, notice the occipital bone had dozens of different characteristics on it. How many of those characteristics are you actually responsible for?
4.
How do you know?
And under occipital, there's 4 listed.
Exactly. And so those are the 4 things that you're going to be responsible for. On the maxillary bone, how many bone features are you going to be responsible for?
2.
2. On the vomer, how many bone features are you responsible for?
On the what?
Vomer. Notice on the previous page, one of the bones that you are absolutely positively responsible for is the vomer, right? How many, well, the one indicates that you have 1 vomer bone as opposed to 2 nasal bones. But how many bone features of the vomer are you responsible for? How many bone features of the nasal bone are you responsible for? Believe it or not, it's not a trick question.
2.
I like the second answer that I got. How about none?
Okay.
All right. Does that mean that the vomer and the nasal bone don't have bone features?
No.
No, they absolutely positively have bone features. In fact, if we go back to that great website we were just looking at, I'm sure if we looked at the vomer, we would see dozens of cool bone features that they would be able to show us on that. But you're not responsible for any of them on the exam. So actually, this is we're already on the right place. So let's do that. So if instead we go to the vomer, right here, we see a bit of the vomer. Here's a nice, right? Notice the vomer's got plenty of bone feature characteristics that you could be held responsible for, but you're not. You don't need to know any of the bone features for them. The only thing you're going to be responsible for are the ones that are on your list. And let's be more precise about what I mean by this. We got to make sure we understand our definitions. On your bone list, and actually, let's do that on your bone list. I like it better there. Let's do it better there. So let's go back here. Let's go back here. Make this large again. And throughout, while we're looking at this, even though it says it right here, let's make sure we understand definitions. When I say a bone feature, what I am saying, what I mean by this is it is a characteristic of the bone. All right. If I'm holding that bone in my hand, like for instance, I have this pen in my hand and this pen has the sharp point to it where the ink comes out, that is a characteristic of this pen. So if I have my bone and it has this little hook piece that comes off of it, that is a piece that when I'm holding it in my hand, that is a feature of the bone. It is a bone feature. I make a point of emphasizing this because there are also things called skeletal features. A skeletal feature, as the name indicates, is a characteristic of the skeleton. So what this is this is a feature, a characteristic that is made of 2 or more bones. So notice I have bone A, which happens to be my right hand, which has a hook. I have bone B, which happens to be my left hand, which also has a hook. But notice when I put those two together, I get an arch. If I'm just holding one bone, do I have the arch?
No.
No, the only way I have the arch is if I put the two bones together. So that
So
makes
would
the arch would be a skeletal feature. So a skeletal feature is made by 2 or more bones and their features that are going together. Now, someone was about to ask a question. What was that question?
sorry. So for example, the one hook would be a bone feature, and then together the two of them would
Yeah.
be a
This
skeletal
would be a
feature.
bone feature.
Okay.
This would be a bone feature. You put them together, you get a skeletal feature. Now, many of the skeletal features you're going to be responsible for are found on the skull. So we call them skull features. So a skull feature is basically just a skeletal feature of the skull, or we could call it what it is, 2 or more bones or bone features. Or let's say it this way, a structure, a characteristic made of 2 or more bones or bone features of the skull. Now, for the skull features, since we're starting with the skull right now, you are responsible for both identifying the skull feature and for identifying the bones and bone features that form them. And how will you know if it's a bone feature you need to know?
On the list.
Exactly. So what I'm going to do is break you up into 9 groups. These 9 groups will be given some of these bones, and we're going to do the axial skeleton first. So that means the skull, that means the vertebral column, and that means the rib cage. And starting next week, we are going to do these presentations. On Monday, my hope is to get through the skull. Let me rephrase that. We will get through the skull on Monday. So you guys will be responsible. Any group that gets a skull bone will be responsible for presenting this on Monday. Now, do you have these bones sitting at home with you? No, of course not. But there are plenty of places where you can go to get the pictures of them. I've made this incredibly simple for you. One of the ways that I've made this incredibly simple for you is if we close this. I guess I have to erase all this stuff now. And we go back to our modules. Notice in our modules under the group lab activities, there are some images for the axial bones. So here you see some pictures of the skull. So again, if you were doing that occipital bone we just saw, you could use this picture from the top. You could use this picture from the bottom. You could use stuff like that to be able to show it. So we have lots of different views of these pictures that you can use. Here's our ethmoid bone, if you're responsible for that, or sphenoid bone, and so on and so forth. These are just random pictures I found on the interweb that you are welcome to use. Yes, Ava, you have a question?
Yeah, so can we make like a presentation slide on like a Google Doc and then share the screen?
Absolutely, that's exactly what you're going to do.
Okay.
You're going to make a Google Doc or a PowerPoint presentation, or you can just show this PDF. If you want a PDF and then you want to be able to draw your own arrows and go, hey, that is the Christagala. Perfect, that is fine. However you and your group want to do it is fine. And again, you don't have to have this stuff memorized. So if you have a piece of paper that has it all written down and you're just reading it off, that's totally fine. Right? You are going to be sharing your screen. You're going to be using these images. And like I said, I've given you images. You're welcome to use these. However, as I also pointed out, there are some amazing images on that. Whoops, hold on. On this anatomy standard. So again, if you're presenting, for instance, the maxilla, right? There's some amazing images, show how it pulls it out of the skull. You get to see it from the front and from the back and from the bottom and from the side and from the inside. And so, I mean, there's some amazing pictures here. There's even some videos that show this. So these are great resources that you are welcome and encouraged to use if you would like. It's not required. You can, if you find your own, or do it the same way I got that list, the same way I got this list of the bones. This one here was just by doing a Google search and finding them. So use your own resources, use the PAL, use the whatever this is, the anatomy standard, but find some pictures that do a good job of presenting the bones for us. You're going to present us the bones and present us our bone features. Now, let's go back to our list here for a second.
As for pronouncing,
I don't care how you pronounce.
Okay.
Again, you're not being graded on your presentation skills. You're not being graded on your pronunciation, right? I am not a good speller. Right? It's even worse when I'm trying to type and talk at the same time. But even when I'm not trying to type and talk at the same time, I am not a good speller. So when I was learning all of this stuff way back when I was in school, I was all saying the stuff phonetically, the way that it looked, because that would help me to remember how to spell it. My biggest challenge when I started teaching was relearning how to say the things the right way, because I'd been saying them wrong in my head for 15 years. So trying to, that was the trickiest part for me. So again, being the smart, sophisticated students, know what you're responsible for. In this class, you're responsible for spelling. I don't care how you say it. So pronounce it wrong all you want. I don't care as long as you spell it correctly on the exam. Okay. Now, you are responsible for presenting the bone and the bone features. If it is a paired bone, oops, what happened there? You need to help us to distinguish left from right. So tell us how you would do that. And all of the bones, you are required to know in an intact skull. So like that occipital bone, you need to show it in an intact skull and be able to identify. However, the following bones, the ethmoid, the sphenoid, the mandible, the maxilla, and the palatine, these 5 bones, you must also show disarticulated. What does that mean?
Pulled apart.
Yeah, which basically means separated from the skull. One last thing, has nothing to do with the presentations, but remember, you are responsible for all the bones. So for instance, as I mentioned, on the exam, can I hold you responsible for the vomer bone in the skull? For the nasal bones on the skull? Yeah, absolutely. Are there any bone features for them you're responsible for? These questions should be getting easier. No, no bone features you're responsible for, but you are definitely responsible for the bones. Okay. Our goal for Monday, as I mentioned, is our groups that are presenting bone features of the skull will present us their bones and present us their bone features. And then together with my help, we will take those bone and bone features and put them together to make the skull features. So all the bones and bone features of the skull, we will then put together to make the skull features. And I'll do that. It's a little too challenging. I'm not going to make a group do that. But your guys are going to give me the puzzle pieces, and then we'll put those puzzle pieces together to make the skull features. Like I said, you are not being graded on your presentation skills. I do not care how you pronounce things. I don't care how you have it. If you have to have your script written down and read it word for word, you are going to do this as a group. I expect everybody in the group to be on camera. I expect everybody in the group to present some part of it. So for instance, if you have the temporal bone and there are 3 of you in your group, then each of you present 3 bone features. You'll all be on camera for the whole time. But just like right now, I'm on camera and no one's looking at me. You guys are all looking at the pictures and reading all the words and doing all that. The same thing is going to be happening during these presentations. Just like when we went over the histology together, the goal of this is to familiarize yourself with the materials, but not master it. So we're going to go through this together, make it an interactive process. The one other caveat that I will warn you about is I will not record these presentations. I am comfortable being on camera. I am comfortable speaking and presenting and then having that available on YouTube, but I know not everybody is as comfortable presenting. So to encourage everybody to be comfortable in participating in this, I will not be recording this for posterity's sake. You will not be on YouTube and become YouTube famous as a result of this. But that also means that you have to pay attention when people are doing their presentations, because it isn't something you'll be able to go back and look at on camera later. All right. One last important point I want to emphasize about this. If we've learned anything, we've learned many things in this class. Animus Hatus is one of the big ones. Pressure is what makes the world go round. But the other thing that we've learned is vocabulary is important. And the same thing is true on this exam. Vocabulary is very, very important. And your book has a nice table that talks about where many of these bone features get their names, right? An indentation in a bone is a fossa, whereas a hole in a bone is a foramen, right? A small, smooth bump that sticks up is a condyle. A large, rough bump that sticks out is a trochanter, whereas a small, rough bump is a tubercle. Can you use those two words, trochanter and tubercle, interchangeably on the exam?
No.
No. So is it important to know these? Yes. Again, is it like a vocabulary quiz where I'm going to ask you the definition of what a trochanter is? No.
No.
But if you know that a trochanter is a large roughened attachment point, whereas a tubercle is a small roughened attachment point, when you're looking at these bone features on the exam, it might help you to be able to tell them apart. So you need to make sure you're using the right words. So understanding the definitions will be important. But like I said, it's not a vocabulary quiz. I'm not going to ask you, what is a smooth, round articulating point called, and you're going to have to know that that's a condyle. So if it helps, because the vocabulary can be overwhelming on this, I encourage you to look at that table in your textbook to be able to figure that out.
I'm a little confused. So for clarification, you're going to be using this vocabulary to describe certain things on the exam, and us knowing that will help our understanding of what you're asking.
Right, because
Okay.
on the exam, I'm going to show you a, for instance, let's find a good example. I'm going to show you a mandible. And one of the bone features on the mandible you're going to be responsible for is the mandibular condyle. But if on the exam, I ask you to identify that structure and you call it the mandibular tubercle, are you going to get credit for that?
No.
No. So knowing that a condyle is an oval, smooth, articulating surface, basically a surface for forming a joint. If you know that that's what a condyle is, and you see on the mandible that it is a smooth oval structure, then you're more likely to remember, oh yeah, that's a condyle. So knowing that vocabulary will help you when you have to identify these things on the exam. Okay. All right, so here is the game plan. We need 9 groups. So let us, oh, here, actually, before I do that, one last thing. Oh, this is where I want to be. In your modules, under the labs, group presentations, notice there are, oh, I lied, 8 groups. I'm going to be dividing you guys into 8 groups. In these 8 groups, group 1, you're going to be presenting the frontal, the ethmoid, and the occipital bone. Group 2, sphenoid, maxillary, mandible, and so on and so forth. So you'll be one of 8 groups. And as I mentioned on Monday, if you are in group 1, group 2, group 3, group 1, 2, or 3, you're presenting on Monday. Everybody, so that group is, those are the 3 groups for Monday. The other groups will be on Wednesday next week. So once I assign you to a group, randomly assign you to a group, these will be, you'll come to this page and this will show you which bones you're going to be responsible for. Now, I'm going to save myself some time by talking to you guys all about this right here. One of the things that I want you to notice when you look at your list of the vertebrae, for instance, notice cervical, thoracic, and lumbar vertebrae all have bodies. They all have spinous processes. However, does the spinous process look the same, do you think, on all 3 of them? No. So your job in those groups are going to be not just to present the bone features that are on the list, but to tell me how the cervical ones are different from the thoracic and the lumbar. There are, for instance, 12 thoracic vertebrae. Are you going to need to tell the difference between the 3rd thoracic vertebrae and the 8th thoracic vertebrae?
Yes.
Okay, I'm mean, but I'm not that mean, right? No, you're not going to have to tell the difference between all 12 of the thoracic vertebrae, but do you need to be able to tell the difference between the thoracic and the cervical and the lumbar?
Yes.
Yes, absolutely. I'm not going to hold you responsible for individual vertebrae, except for two. Two of the cervical vertebrae are very, very highly specialized cervical vertebrae that allow us to do some very, very special stuff. So one group is going to be responsible for presenting to us what a typical cervical vertebrae is. And a different group is going to be presenting those two special vertebrae. These two special vertebrae, C1 and C2, are so special they get their own names, Atlas and Axis. And they are unique, they are distinct, and those are the two special vertebrae you will need to know. So what will happen on Wednesday is our cervical group will go first, showing us what a typical cervical vertebrae is. And then group 7, that's given us the special vertebrae, will go next to show us that this is what they typically look like, and these are the two special ones. Okay? So for the vertebrae, tell me the difference between these, and then there are only two unique ones you need to know. All right. That is the game plan. Any questions on that? All right, one last thing. I do have one student who reached out to me. She had a doctor's appointment for her baby, and so she wasn't able to be here right now. At least I believe she's not here right now. So one of the groups that's going to go on Wednesday, because I'm not going to force her to go on Monday because she'll miss this, but I will be adding one additional student to a group. I will warn you about that ahead of time to let you guys know that. And it'll be one of the Wednesday groups, just to let you know. So whatever group has that, I will do that with. But so other than that, everybody who's here is getting into a group. I need 8 rooms. Assign automatically. Perfect. So excellent. All right. So any other questions?
Could we in advance send you like our slides and have you look over it to make sure we're not missing anything?
No.
Okay.
You have your list that tells you. Yes. So when you get into a room, whatever room number you're in, that is your group number. So here's what's going to happen. I'm going to send you to your groups now. Once you're in your groups, go to this page and see what bones and bones you're responsible for. Then look at your list of bone features and then start working as a group. I've already set it up so that once you're in groups, you guys can share your screens so that you can do this. Which reminds me of one other thing. I will also be setting up discussion boards for you. You have the rest of the class time, which is about an hour and a half to work together as a group, but that may not be enough time to finish all this information. I encourage you, the first thing to do, that if you'd like to share phone numbers or email addresses or things like that. However, if you're not comfortable sharing those things, or again, your schedules are so opposite of each other, because after all, this is just randomly being assigned. If that's the case, then I will be setting up a discussion board where you're able to communicate asynchronously. So you can use that discussion board to share images or share files and do things that way as well. All right. All right, excellent. Then I will open the rooms for you. The rest of the class time is yours. Use this to communicate, discuss that. I will be here in the main room. If you have any questions, you can come back here and ask those questions. But the rest of the class time is yours to do with as you like. So any questions before we break?
Are we meeting at the end, or are we just leaving after that 12:30?
No, when your group is done and satisfied,
Okay.
you are welcome to move on your merry way.
Great, thank you.
In fact, if after 5 minutes you realize you all live within a block of each other and you want to go meet at Starbucks and do it there instead, you're welcome to do that as well. The rest of the class time is yours to use as you need to use it. All right. Any questions?
I have a question.
Yes.
So basically all we're doing is, so we're obviously going to present the slides and go over the features of each. That's it. Or what else are you expecting of us?
That's it.
That's it? Okay.
Yep. Right. You're going to have a picture of a bone and you're going to say, this is my bone. It is an iPhone. Right. Here is the camera. Here is the button that starts it. Right. Here is the volume control. This one makes it up. This one makes it down. Right. As you can see, mine is in an OtterBox because I'm a klutz and I drop it all the time. Right. So you're going to basically just present that with an image on screen that you're sharing And that's it. It's really, like I said, this is as low key of a presentation as you will ever have to do in your entire academic career. The whole point of this is just to make a boring process of this is a bone and this is the head and this is the tail and this is the whatever, a little bit more interactive to make it a more interactive learning process. Yes, everybody needs to be on camera in your group. So if there's 3 of you in your group, if Ava, me, and Amanda are a group, then we will all be on camera while we are presenting. Ava can go first, and then me, and then Amanda. And again, as long as you're all on camera and you're all active in the process, even if you are confused and get things wrong, you're still going to get full credit. You're not being graded on your presentation skill. Really, the grade you're getting for this is a participation grade. As long as you're active in the process, then you're going to get full credit for it. Sarah, did you have a question as well?
Yeah, so when, like, say it's my turn to do the presentation, do I need to create my own presentation slide for my particular bones that I'm going over, or can we make one big one for the group?
It's totally up to you. If you guys want to have, if Ava's more sophisticated and she's going to set up the Google and we just send her the pictures and she puts it all together and she presents it and goes through the slides while you and I and her talk, that's fine. Or if each of us want our own pictures to present our own portions of it. Now, again, if you think about it, if the 3 of us together, and again, let's move to the appendicular skeleton. If we're doing the femur, which has a ton of bone features on it, it's silly for us each to have our own picture of the femur. So it kind of makes more sense to have each person do their own thing. But yeah, however you guys want to do it as a group is fine. It's easier if you have one picture because you don't have to bounce back and forth between everybody, but it's not required. If you want to do your own part and do it all separate, then that's fine as well.
Okay.
Yep, there's no wrong answer for that. As long as all 3 of you are on camera and all 3 of you are presenting, or 2 for the groups that have 2, then you're doing it right.
Okay.
All righty. Excellent. Any other questions?
You said groups 1 through 3 are going on Monday, or 1 through 4?
1 through 3. Groups 1 through 3 will be going on Monday. And so they'll be the ones that will, break the seal on the process and show the rest of us how it's done so that Wednesday it'll go even easier. Okay. All right. I'm going to break you into groups. Right now, I will tell you group 5, I'm coming to your room because you're going to be the group that gets the extra students. So I will talk to you guys. I'll come to you and talk to you guys in just a second. All right. Any other questions? All right. Then the lab is yours. Hey, guys.